Alright, welcome to this lesson with me, Teacher Kamanga. Alright, now, in today's lesson in biology, we're going to look at enzymes. So, enzymes, it's a very, very interesting topic that we mostly going to be get interested in because when we understand fully about enzymes, we understand what normally happens in our body. Okay, so by definition, we're saying an enzyme is a biological catalyst. That is very, very simple and straightforward. Now, what we need to understand is that a catalyst is a substance that speeds up the rate of a chemical reaction without itself being changed by any reaction. So what we expect is that if an enzyme has been involved in a chemical reaction, at the end of the reaction, the enzyme will not be changed to just remain the same. So this is very, very important to understand. Now, these enzymes that we're talking about, enzymes are the ones that are responsible of catalyzing chemical reactions in living organisms. So in living organisms, there are a number of processes that takes place. So those enzymes are the ones that are responsible to work inside of these living cells. So now these enzymes that are responsible to work inside of living cells, they are called the intracellular enzymes because we understand that they work inside, so it's intracellular enzymes. Then also we have other enzymes that work outside living organisms, so these are called extracellular enzymes. So we should be able to show the difference between the two. Now, what is important is that we need to understand that substance in which these enzymes they act on or they work on or they work from, they are called the substrates. So we need to understand subscript. So these are substances which enzymes are going to act on. So if an enzyme is acting on a particular substance, that substance is called a subscript. Okay, that is very, very important to understand. Now, what we need to understand is that the enzymes, they are going to fit in the subscript. So now, during the enzyme catalyzed reaction, that enzymes, when it is fitting into that subscript, that part where it is fitting in, it is called an active site. So you're going to see even from the diagrams that we're going to post there, you see how the enzyme will fit in the subscript and the active site is going to be formed. Okay, that is very, very important to understand. Now, the same enzymes that we were probably talking about, uh, what we need to know is that these enzymes, they catalyze reaction forward and also backward. So the reaction of an enzyme can be reversible, can be forward reaction and also backward reaction. Now that said, let's look at some of the characteristics of an enzyme. Some of the characteristics of an enzyme. So to start with, an enzyme is protein in nature. So most enzymes are protein in nature. That is their characteristics. And also another characteristic is that based on the definition of said, these enzymes are biological catalysts. So we understand that enzymes are catalysts. So that qualifies to be their characteristic to say these enzymes are catalysts. Okay. So again, if you remember what I said on the reaction, we said these enzymes, they catalyze both forward and also reversible reaction. So this means also it is their characteristic to say they can catalyze forward and also backward reaction. Another interesting characteristic of the enzyme is that these enzymes are specific. So what we mean is that the enzyme can only catalyze or can act upon one subscript. This is very, very clear. An enzyme can catalyze upon one subscript, meaning that enzymes are specific in terms of their reaction. So we need to understand that. Okay. Another characteristic is that these enzymes are affected by the temperature. They are also affected by the pH. They are also affected by the subscript concentration. The enzymes also are affected by enzyme concentration, and also they are also what we call the inhibiting factors and also the cofactors. So these are some of the characteristics that we need to understand that enzymes will probably portray as they are able to catalyze reactions. Okay, so that is very, very important that we should be able to understand. Now, if we're talking about enzyme being specific, enzymes, they work under what we call the lock and key mechanism or the lock and key hypothesis. We understand that one key will only open one lock. This means that only an enzyme will act on a specific substrate. For example, if an enzyme, it can work on proteins, that enzyme will not work on starch because it can only work on what? proteins. So we need to understand, because it is not going to fit in the subscript where it is going to work from more. For instance, the example that I've given on starch. So we need to understand. Okay. Now, when we come to how temperature can affect an enzyme activity. Now, what normally happens is that the enzyme activity increases with the increase in temperature. This is very, very important to note. The enzyme activity increases with the increase in temperature. Now, this increase can only increase up to what we call the optimum temperature. The optimum temperature, it is the temperature at which the enzyme works best. So we understand that when it reaches that particular temperature, then the rest of the reaction, there will be what we call denaturation. So now after the optimum temperature, the rest of the reaction, the enzymes becomes denatured and loses its catalytic function. So meaning that that ability of it catalyzing a subscript will be lost because it has denatured. Now, when we're talking about denaturation of an enzyme, we talk about an enzyme losing shape, failing to fit. For instance, if we're talking about a lock and key, you're going to realize that if you were to hit a key will not fit in that particular lock. This is just an example if an enzyme has been denatured. So the enzyme will not fit in that particular subscript. So we need to understand to say temperature plays a very, very important role in this case. Another factor that we've talked about, it is the pH. pH concentration. So when we're talking about the pH, we're just talking about the measure of how alkalinity or acidic a substance is. For instance, when you're talking about the pH scale, it ranges from 0 up to 14. So you're going to realize to say from 0 up to 6, that is acidic environment. Then we have 7, which is neutral. Then from 8 up to 14, that is alkalinity. So you're going to understand, say, there are some enzymes that can work best in acids, and there are some enzymes that can work best in alkaline conditions. So for instance, if we talk about protein, the enzymes that can catalyze on protein, they work best in acidic environment. That's why you're going to realize that protein digestion, it mostly starts in the stomach by the enzyme known as pepsin and also renin, because those, they work best in an acidic environment. While when we talk about starch digestion, which starts from the mouth, and part of it is characterized by the salivary amylase, which the enzyme works best in the alkaline condition. So you're going to realize that the enzyme that works best in alkaline condition cannot work in an acidic environment, and the one that works in an acidic environment cannot work in the alkaline condition. So we need to understand to say the pH is very, very important to understand, because it can affect the rate at which this particular enzyme is going to work best. So we need to understand. Now, the other factor that we need to understand is that the pH value at which this enzyme works best is also known as the optimum pH. So you realize that at the peak of a graph, that's where we're going to locate the optimum temperature, and also that we're going to locate the optimum pH. So when you just look at the climax of the graph, you'll be able to locate that. Okay, that is very, very important to understand. All right, now we also try to look at the naming of enzymes. Now, one way of naming these enzymes, they use the first part of the subscript name and the suffix. So the suffix in most cases, it's ASE, as, ASE. So for instance, if for example you are talking about starch, let me say starch. Okay, so starch in most cases there are two enzymes that we need to talk about. There is what we call the amylose and also the amylopectin. So these are the two enzymes that we are going to talk about for starch or carbohydrates. So now, when we're talking about the maltose, we're talking about the sucrose, we're talking about the lactose. All those are the ones that are responsible for the digestion of starch. Now, when we also talk about these type of sugars that we've just talked about, like maltose, sucrose, and also lactose, if we were to name an enzyme that is going to catalyze, maybe for example on maltose, we'll only put the suffix there. I hope we get that one. So for example, we have amylase that is responsible for starch. Then also for maltose, we have what we call maltase. So where there is the maltose, where there is OSE, we remove, we put ASE. Then that will be the enzyme that is going to catalyze on that one. So for instance, again, when we're talking about the proteins, we have what we call peptides. This is a form of protein. So when we're talking about an enzyme that is going to catalyze on these peptides, we just remove the IDE, then we put the ASE, which enzyme will be what we call the peptidase enzyme, which is going to catalyze on that one. So we need to understand to say this is how the enzymes in most cases are being named. All right, so that is very, very important. Now, these enzymes are also very important in our daily lives because they are used in industry. So, for example, enzymes can be used in making the biological detergents. So these detergents that we use, they need to put enzymes that are going to catalyze on the food substance that is probably for on our clothes as we are eating, so that when washing it is going to move away. We also have breweries and also baking, they use enzymes. In also making sweeteners for food and also drinks, they use the same enzymes. In also the dairy industries, these dairy industries that we're talking about, mostly they use the renin, which is used as a coagulating to coagulate milk. Most when we're talking about digestion, we talked about the renin and also how it coagulates the milk. Well, enzymes also are used in tanning of leather, extraction of processing of fruit juice, and also tenderizing of meat. So we need to understand to say these are some of the use of the enzymes. Okay, so now what we need to understand is that we need to familiarize ourselves with number one, the definition. We also need to familiarize ourselves with number two, the characteristics of the enzymes, and also we look at the factors that can affect the activities of an enzyme, and also we looked at, or have also looked at how an enzyme is named, and also have looked at what are the uses of enzymes in industry. So these are the major things that we're going to look at in most enzymes. So pay close attention to the video as it is been explaining how we are going to calculate, how we're going to find the answers, and also how we're going to analyze questions based on enzyme.